Chapter 4 is divided into two sections. Section 2 is on the permeability of the plasma membrane. We looked at the structure of the plasma membrane in 4.1, so let's look at the permeability of it. It is called differentially permeable. That means that some things can go through and some things can't, and the factors that determine how a substance can be transported across the membrane depends on the size of the substance and the polarity or charge of the substance. Now we're going to talk about the concentration gradient. More of a substance on one side than the other is what makes the concentration gradient. And let me give you a practical thought to put in your head about this. So let's pretend that we're going on a float trip. We're going to relate everything back to a float trip. So the concentration gradient is the direction that the water is flowing in. So going down the concentration gradient is an area from higher to lower, like going down the river. Going up a concentration gradient is going to be going from an area of lower to higher, which would be trying to get into the canoe and actually paddle upstream against the current. This does require input of energy, which makes sense when you think about using a canoe and paddles and trying to go up the current. Some molecules move freely across the membrane, such as water molecules, small molecules. If they're not charged molecules, they go quickly and easily across the membrane. Water can also use what's called aquaporins to cross the membrane. Other molecules cannot. They are going to use the protein channels that we talked about yesterday, the carrier proteins that we talked about yesterday, and vesicles. And those vesicles are going to be formed through the process of endo or exocytosis, depending on the direction the molecules are going. So if we look at this picture with the plasma membrane, we see the proteins inside the membrane, and these are integral proteins. We have them on both sides. And non-charged molecules are going to go through. Macromolecules are going to be harder to go through. We're going to have endo and exocytosis for those. Water can go straight through. Charged molecules and ions don't easily go through, and we'll talk about how to get those across the lipid bilayer. Let's take a look at this table, which is very important. Passage of molecules into and out of the cell. The ones that do not require energy, the processes that don't require energy are diffusion and facilitated transport. The reason they don't require energy is because of the direction that they go. They go from a higher to lower concentration. So they are going to go with the concentration gradient. So if we're looking at energy not being required for diffusion and facilitated diffusion, what we're talking about is getting into the canoe and going downstream with the flow of the current. Okay, so we have to have the concentration gradient going from higher to lower. Examples are lipid-soluble molecules, water, and gases. For the facilitated transport, it's going to use not just the flow, but carrier or channel proteins that help them go across the concentration gradient. Some sugars and some amino acids use these. So these don't require extra energy because we're going with the flow, like flooding downstream on a float trip. However, if we use active transport, exocytosis, or endocytosis, it's going to require energy because we're going against the concentration gradient. So energy is going to be required going toward a higher concentration gradient. Sugars, amino acids, and ions are examples of active transport. We're going to get into these a little bit more as well. Exocytosis is going to take and form a vesicle, and it's going to push substances outside the cell. When the vesicle fuses with the plasma membrane, and we get macromolecules out of the cell that way. Endocytosis is going to go toward the inside of the cell, forms a vesicle, and macromolecules, again, are what kind of molecules that we're going to talk about getting into the cell. Okay, diffusion, movement of molecules from an area of higher to lower concentration. You must memorize this definition. It's very, very important that you do. This goes down a concentration gradient because it goes from higher to lower, so it's going on the float trip. A solution contains a solute, which is a solid, and a solvent, which is a liquid. This is a picture of what would happen if I dropped a crystal of dye in the water, and you've done an experiment like this before, part of a lab where you put food coloring last year in Chem 1, food coloring into water, and you watched it diffuse. So in this case, the water molecules are the solvent, the dye molecules are the solute. They're going to go from an area of greater concentration to an area of lesser concentration until we reach equilibrium, and they're evenly distributed throughout. Gases can diffuse through membranes too, and if you take anatomy class, we'll talk more about this. Oxygen and carbon dioxide enter and exit by diffusion through a membrane, at the alveolar level here inside the lung. Several factors influence the rate of diffusion. So what can speed up or slow down diffusion? Temperature is key, because if you increase temperature, you increase the rate of diffusion. And if you think about this, if you're going to put sugar into tea, it's going to dissolve quicker in hot tea than it is in cold tea, because sometimes you can add packets of sugar and add 10 times as much sugar and still not have the flavor because it doesn't diffuse throughout nearly as quickly. Pressure is another one. Electrical currents and the size of the molecules are going to influence the rate of diffusion. Osmosis is a special case of diffusion, and you need to memorize this definition. It's the diffusion of water across a differentially permeable membrane. So this is movement of water from an area of greater to lesser concentration because it's diffusion across the membrane. Diffusion always occurs from higher to lower, and we're going to talk about osmotic pressure. Two, it's the pressure that develops in a system due to the flow of osmosis, or the flow of water across the membrane. The greater the possible osmotic pressure, the more likely it is that the water is going to diffuse in that particular direction. All right, so we look at this setup here, and this is a solution with a 10% Kool-Aid, we'll say, solution. And out here we have a 5% Kool-Aid solution. And the membrane is not permeable to the solute. So these green dots over here are not allowed to go back and forth because they're too big. So this is called the thistle tube, and here's our membrane down here, this red line, and this is the beaker. Okay, so the 10% solution is going to have less water than the 5% solution. 95% water here, 90% water here. Water is going to move from an area of greater concentration, which is the 90% here, to an area of lesser concentration, which is going to be into this thistle tube. Now the water can move across the membrane, but the solute can't. So what happens after a time is you'll notice that the water is going to go from 5% solution, which 5% solute solution, so 90% water to 95% water to 90% water, and you notice the increase in the thistle tube of the water. That dilutes the concentration of the solute as well. Okay. Now we have a concept that's kind of hard to grasp, so you just have to memorize, and that helps, and then we'll do some applications. So isotonic, this means that the solute concentration is equal inside and outside of the cell. Hypotonic, it's a solution that has a lower solute, hypo is low or below, concentration than inside the cell. And a hypertonic, hyperactive, someone who is above active, normal active, is a solution that has a higher solute concentration than inside of the cell. One thing to keep in mind as we go through these iso, hypo, and hypertonic solutions is that everything is relative. For example, in this case, it says the solute concentration is equal inside and outside the cell. Hypotonic has a solution has a lower solute than inside the cell. So we're talking about the solution in respect to the cell. Hypertonic has a higher solute than inside the cell, so it's in respect to the cell. So you have to be careful about the wording when we talk about these. All right, isotonic solutions. When you have an isotonic solution, there's no net gain or loss of water. For example, if we have this cell here, this animal cell, and this example of this would be a blood cell, and we see this size of the blood cell is much smaller. In an isotonic solution, there's no net movement of water. By net movement, we mean that water is moving back and forth, but there's not a shift to more water being outside or inside the cell because the overall solution is equal on both sides, so the water is going to go back and forth at that rate because they're already at the same concentration. Okay, in a hypotonic solution, the cell is going to gain water. The reason it's going to gain water is that the amount of solute, it's a hypotonic solution, so the amount of solute out here is less than what's in here. So let's just say it's 5% out here, 10% in here. That means the percentage of water is greater outside, which is at 95%, than inside, which is at 90%. Remember that water is going to go from an area of greater concentration to lesser concentration until equilibrium is reached. So the water is going to go from the outside, which is at 95% in this case, to inside, which is 90%, and it's going to cause so much pressure inside the cell it could cause it to burst, or the process is called lysis. Let's look at hypertonic solutions. In this case, the cell is going to lose water. So in a hypertonic solution, the percentage of solute is greater outside. So let's say the percentage of solute out here is 10%, and the percentage of solute inside is 5%. That means that we're 90% water here and 95% water in here. Water is going to move from there greater to lesser concentration. So it's going to go from the 95% water to the 90% water, which is going to cause the cell to shrivel. That's called crenation. Same thing happens in a plant cell. Turgor pressure is a word that we talked about in class, and turgor pressure is how much pressure is exerted on the cell walls, mainly due to the amount of water inside of the central vacuole. So if you have an isotonic cell, there's no net gain or loss of water. So these are plant cells, and all these little green dots here are the chloroplast, and they're full. You notice the chloroplasts are taking up the whole entire cell. Again, a hypotonic solution is going to cause the cell to gain water. And you see this here. You notice that in the pictures of these cells with the plant cells, we have a rather large clear area here, just meaning that central vacuole is just has so much water pressure inside of it, it's pushed all of the chloroplast around the outside edges. That helps keep the plant cell full of water and the turgor pressure high so that the plant is upright. Hypertonic, on the other hand, is going to cause the cell to lose water, and that's what we see here. So if you look here, you'll notice that so much water has been lost that the cytoplasm is kind of shriveled up here, and all the chloroplasts are centralized in the center, and that can cause a process which causes the cytoplasm to shrink, and we call that plasmolysis. And that's just a picture that shows you those side-by-side animal cells and plant cells. Okay, transport by carrier proteins. Carrier proteins are specific. They combine with a molecule or an ion to be transported across the membrane. Carrier proteins are used for facilitated transport, active transport, and we're going to look at pictures and examples of each one of those. To facilitate means to help. So facilitated transport is a helping process. Small molecules that are not lipid soluble may need help across the membrane. Molecules that follow the concentration gradient may also need them, and energy is not required for this. So this carrier protein is just going to help them get from an area of greater concentration to an area of lesser concentration where it needs to go. It goes with the concentration gradient. This is going to have to wrap up 4.2's first section, and there's going to be a second video that's going to cover active transport and endo and exocytosis.